Today, we're going to continue on with genetic mutations and disorders. First, of course, I want you to go into the top of the Google Classroom on the top of the unit and submit your daily attendance. Yesterday, day 1, we went through slides 1 through 15. Today, I want you to review slides 16 through 26 if you haven't already, and we will be reviewing those slides. Let's go to slide 16. Yesterday, we talked about specific disorders and mutations linked to specific genes. Today, we're going to be talking about chromosomal disorders, mutations and disorders linked to entire chromosomes. And these occur due to failures in meiosis, non-disjunction, where a mistake in meiosis where chromosomes do not come apart. Right? You can see in the picture, metaphase chromosomes are lining up on metaphase 2 in meiosis, but they do not, this one chromosome does not come apart at the centromere, and that results in a chromosomal disorder. In examining chromosomes, an important topic to cover is karyotyping. Karyotyping is a full picture of an individual's chromosomes, right? We have 46 chromosomes. We have a male karyotype and a female karyotype. Chromosomes are sorted in number 1 being the biggest, and they decrease in size. The same with female. The only difference being is that male have one X and female have one Y. Male have one X and one Y, and female have two X's. But karyotyping is important because geneticists can look at the possibility of genetic disorders in people who wish to have children, especially as people get older and wish to have children. They can weigh the risks of having a baby, knowing their chances of having a baby with a particular genetic disorder, weighing the risks before the woman gets pregnant. But let's stop and check. How many total chromosomes is a person supposed to have? So yes, a person has 46 chromosomes, 23 pairs, as you can see in the karyotypes. 44 of those are autosomal chromosomes, and 2 of those are sex chromosomes. Examples of chromosomal disorders. Down syndrome. So now, like I talked about how humans are supposed to have 46 chromosomes, that doesn't always happen. So, in Down syndrome, also known as trisomy 21, people with this syndrome have 3 copies of chromosome 21. So, that means that they have 47 chromosomes instead of 46. Down syndrome results in physical growth delays, intellectual disability, and characteristic facial features. It's important to note also that the risk of Down syndrome increases for mothers, expectant mothers, as they pass 35 years of age. Now, Turner syndrome. We see a karyotype for Turner syndrome. Turner syndrome is caused by non-disjunction of the X chromosome. Therefore, it only affects females, and they only inherit one X chromosome. So, instead of having 2 X chromosomes, they only have 1. So therefore, their total number of chromosomes is 45. And as a result, they do not fully develop at puberty, and they are unable to reproduce. It means they can't have children. Now, if we look to a chromosomal disorder that only affects males, we see Klinefelter's syndrome. The karyotype is shown. In this disorder, males, through non-disjunction, inherit an extra X chromosome. And yes, it only affects males. This results in these men being tall, but not muscular, and they also do not fully develop at puberty, and they cannot reproduce. So they will have two X chromosomes. They will have XXY. So they will have 47 chromosomes total. Now, let's stop and check. While humans are supposed to have two sex chromosomes, either XX in females or XY in males, is it possible to have different variations or numbers? Yes, so of course. So we just talked about several different syndromes, right? Klinefelter's syndrome, where you have three sex chromosomes. Turner syndrome, where you have two sex chromosomes. And there are also other conditions with incorrect sex chromosomes, but we're only going to explore a few, like Turner's and Klinefelter's. Now we're going to move on to pedigrees. What is a pedigree? A pedigree is simply a chart that shows the presence or absence of traits according to a familial relationship. A familial relationship is just the genetic relations between family members and offspring. Based on the analysis of pedigrees, it's possible to determine if an allele is for a trait is dominant, recessive, autosomal, or sex-linked. Remember, we talked about alleles. Every gene has two alleles, and they can separate, and when reproduction occurs, they join together from the mom and the father and create that trait. And dominant traits have dominant alleles, and they are dominant over recessive. So recessive alleles, you need two copies of recessive alleles, remember, and you only need one copy of a dominant. And if we're looking at sex-linked or autosomal alleles, so remember we have 44 autosomal chromosomes, and so genes and alleles are located on those autosomal chromosomes, or there are some genes and alleles located on our sex chromosomes, which I say usually in males it's XX, and usually in males it's XY, and usually in females it's XX. Here's an example of 4 different pedigrees. We have an autosomal dominant condition, so it is linked to an autosomal chromosome. So the black represents affected individuals, so the mother and father are affected, and so the mother is a circle, the father is a square, and the son is affected, but the daughter is not affected. So we can interpret this as the disease cannot be recessive, as two affected parents could not have an unaffected offspring. So therefore, the parents must be heterozygous. They have a dominant and a recessive allele. Let's examine autosomal recessive again on one of the autosomal chromosomes, but recessive. Both parents are not affected. The mother's not affected, the father's not affected, the son's not affected, but the daughter is affected. She has the disease. So it cannot be dominant, as two unaffected parents could not have affected offspring. So the parents must be heterozygous, meaning that they're not affected, they have the dominant allele, but if you remember in the Punnett square, there's always the probability of those two recessive alleles that the parents carry joining together and that's why the daughter has the condition. Now let's look at sex-linked. X-linked dominant. So we have several generations. We have a parental, remember? We have our F1, and we have our F2. Originally, the mother is infected, or has inflicted with the disease. Her son is, and the son reproduces, and we see that only the daughters are affected. So 100% of incidents of affected daughters from an affected father suggests X-linked dominance, but sex linkage cannot be confirmed. In X-linked recessive, we start out with our parental generation. We have the father has the disease, which is passed on to the daughter, and the daughter then reproduces with a male that does not have the disease, and 100% of the sons from the affected mother have the disease. So that suggests X-linked recessive. Let's stop and check. So why would 100% of male offspring be affected from a mother with an X-linked recessive disease? Remember, males usually only have one X and one Y chromosome. So yeah, the reason would be because the males only usually have one X and one Y chromosome. So if the recessive allele for the disease shows up on the X chromosome, it's automatically going to be expressed because they don't have a second X chromosome, they just have a Y. And that Y does not carry that disease allele, so the X will express it. So that's unfortunate for men. Why they are more likely to have X-linked recessive diseases. Now let's take a look at a specific pedigree for a specific condition. Colorblindness is X-linked recessive, so it most often affects males. In this pedigree, circles are females, squares are male. And yeah, so this is X-linked recessive. The half moon females, right, represent carriers. So while they have the colorblind allele, they also have the dominant normal vision allele, so it doesn't affect them. However, they can easily pass colorblindness onto their sons. Let's look at another pedigree for hemophilia, which also affects males more as it's X-linked recessive. Again, circles are females, squares are males. Shaded squares are hemophilic males. Shaded circles are hemophilic females. And again, yes, so men are more likely to be affected. And a little history on hemophilia. It's basically just the inability of blood to clot. So you could bleed out just by a cut or a nosebleed. Your blood can't clot. So it affected generations of royals in Europe in the 1800s and 1900s. Inbreeding didn't help. It made the problem worse. Cousins marrying cousins. They tried to preserve this royal blood that didn't exist, and they ended up hurting themselves. So let's stop and check. What does a pedigree show? And last, we're going to cover gene expression. What you need to know about gene expression is that certain parts of DNA are turned on and turned off based on cell location. This results in differentiation, how cells become specialized into different structures, different parts of our bodies. And that will end our lesson today. Thank you.